Hi micro folks. This will probably be our last lab video of our coronavirus files. So again, this is in response to the campus being closed down because of the coronavirus pandemic. So hopefully we will be back before the end of the semester to do some of these cool exercises. But just in case we aren't, wanted to make sure that you saw how to do them. So folks, this will be Metabolic Test Part 2. This is in our lab manual, Chapter 17. And this covers a lot of metabolic tests. We're going to be doing the IMViC test, which is the indole methyl red Voges-Proskauer citrate test. We're going to do big butt TSI slants, stab and streak. TSI stands for triple sugar iron, and it's really cool media. It has 3 sugars, glucose, lactose, and sucrose. It has a pH indicator, phenol red, so we'll be able to determine if the sugars are fermented. It has sulfates as alternate terminal electron acceptors and anaerobic respiration, so we'll get to see if our microbes make hydrogen sulfide. It also has sulfhydryl-containing amino acids like cysteine, so we'll see if our microbes can make hydrogen sulfide by the breakdown of cysteine. And finally, we'll be able to determine if our microbes make gas during fermentation of sugars, so that would be carbon dioxide and or molecular hydrogen. We'll determine that by looking for cracks or bubbles in the agar. And the last test we'll do, folks, will be the catalase test. And you might recall from lecture, catalase is a protective enzyme that breaks down hydrogen peroxide into water and oxygen. Okay, folks, so the first test we're going to do are the MViC tests. And the MViC tests, folks, there's a table, and this is on page 182. There's a table comparing the results of different microbes when we run the MViC test. The MViC test, folks, it's actually a series of 4 tests. The indole test to determine if a microbe makes tryptophanase, the methyl red test, which determines if a microbe carries out mixed acid fermentation, the Voges-Proskauer test, which determines if the microbe carries out 2,3-butane-diol or butylene glycol fermentation, and then the citrate test is to see if the microbes can use citrate as their sole source of carbon and energy. So the 4 tests are often run together to help distinguish E. coli, which is an indicator of fecal contamination, from its close cousin Enterobacter aerogenes. E. coli lives in our intestine, as does Enterobacter aerogenes, but Enterobacter aerogenes might also live out in nature, maybe in decomposing organic matter. We say that E. coli indicates fecal contamination because we know wherever feces goes, there'll be a lot of E. coli. And so if we find food or water that has high numbers of E. coli, we really worry about fecal contamination and that there might be any fecal pathogen present, and we know that's not good. In contrast, if we were to only find Enterobacter aerogenes in food or water, as we mentioned, Enterobacter might be in feces, but it also might be found in dead, decaying material, so it wouldn't necessarily indicate fecal contamination. Okay, folks, so what I'm going to do is I'm going to show you how to inoculate the MViC tests using E. coli. And what we would do, and what I'll do after the video, is I will also inoculate Enterobacter aerogenes in the 4 MViC tests. So this weekend when we come in, we'll pull our results and we'll be able to fill in this table, which is on page 184, comparing the results of E. coli and Enterobacter aerogenes. And we're hoping they will be opposite results. We're hoping our E. coli will be indole positive, methyl red positive, VP negative, citrate negative, and we're hoping our Enterobacter aerogenes will be the opposite, so indole negative, methyl red negative, VP positive, and citrate positive. So let's first just show you how we would use aseptic technique to inoculate those tests. Okay. Pages out of here. So again, folks, I'm just going to show you using E. coli, but we would run these same tests using our Enterobacter aerogenes. So the first test, folks, we're going to demonstrate is how to run the indole test. And back in the old days, you'd have to inoculate special indole media. It's a liquid medium that has a high concentration of the amino acid tryptophan. So the question we're trying to answer in running the indole test is, does our microbe make the enzyme tryptophanase to break down the amino acid tryptophan into indole and other end products? So what we're going to detect is actually the end product of that breakdown. We're going to be detecting indole. And this is really neat, you guys. Nowadays, all of our reagents are in these dry slides called indole dry slides. And so real simple benchtop tests, all we have to do is harvest our cells from our pure culture. Sorry, there's a lot of background conversation. Everybody's here trying to get ready for our switch to online classes. So there's a lot of stuff going on. Okay, so I want to make sure that I sterilize my loop. Okay. Because we always use aseptic technique. I touch my plate to make sure it's not too hot. Okay, make sure it doesn't hiss and spit. And I'm just going to harvest some of the E. coli from my plate. And then it's real simple, folks. I just smear my E. coli onto my indole dry slide. And I'll hold this up here. So I'm just smearing my E. coli onto my indole dry slide. And check that out. You guys see it's turning pink or red. That's a positive indole test. And a positive indole test tells me that my E. coli makes tryptophanase, breaks down tryptophan, producing indole. And that's what these reagents. So red or pink is a positive indole. If I tried a different microbe and inoculated the indole dry slide and it didn't turn pink or red, that's a negative indole test, and it tells me my microbe doesn't make indole. Okay. All right. So now we're going to go back and we're going to continue with our indole tests. So we're going to continue, with the MViC test. So I is for indole, M of MViC is for methyl red, and methyl red is a test to determine if your microbe uses a specific fermentation pathway called mixed acid fermentation. And mixed acid fermentation, as the name suggests, you get lots and lots of different acids made at the end of the fermentation pathway. And because you get all these acids made, the pH is going to be really low, like below pH 4.4, 4.5. So the way we run this test, I've sterilized my needle. I'm going to harvest my E. coli. And I'm going to inoculate methyl red broth. And methyl red broth is broth that has lots and lots of glucose in it. Lots and lots of glucose in it. Because we want our microbes, if they can ferment the glucose, to make lots and lots of acids to drop the pH down below pH 4.5. Okay, so I inoculate. I want to check just to make sure I have, a loose cap so it can vent. Okay, and then I'm going to re-sterilize. So, folks, we're going to let our microbes grow for 48 hours. And then this weekend when we come in, we'll do a movie. I'm going to add the pH indicator methyl red. And the reason we can't add methyl red ahead of time is it would kill the bacteria, and we have to have them grow and ferment before we can run the test. So with methyl red will turn red at really low pH, below pH 4.4, 4.5. So if our microbe carries out mixed acid fermentation, we add the methyl red pH indicator, it should turn red. So red is a positive methyl red test, telling us our microbe performed mixed acid fermentation. If the microbe didn't perform mixed acid fermentation, the pH won't be low enough to turn the methyl red red, and instead the methyl red will appear yellow or orange, right? So again, red is a positive methyl red test, not red is a negative methyl red test. In the MViC test, the third test for V is called the Voges-Proskauer test. And this is a test to see if our microbe uses a different fermentation pathway, the 2,3-butane-diol fermentation pathway, or butylene glycol fermentation pathway. And so it's in some ways similar to the methyl red test. Okay, we're gonna, I'm gonna harvest my E. coli. Okay. I'm going to take my VP broth, and this is actually the same broth as methyl red, just a high glucose broth. Okay, I'm going to inoculate. Make sure I have a loose cap. Incubate 48 hours. And then after 48 hours, we're going to add 2 reagents, alpha naphthol and 40% potassium hydroxide. If our microbe use butylene glycol fermentation, there'll be this unique intermediate called acetylmethylcarbinol produced, or acetoine. And when we add our alpha naphthol and 40% potassium hydroxide, we'll get a red color. So again, red is positive for a VP test, meaning that our microbe performed butylene glycol fermentation. If there's no red, it's a negative VP test, and we know our microbe didn't carry out butylene glycol fermentation. Okay, folks, the last test of our MViC test, the C is for citrate, and this is a test to see if our microbes can use citrate as a sole source of carbon and energy. Okay. And the citrate test we use, folks, is this beautiful Simmons citrate auger. It has this really cool pH indicator called bromethymol blue. Bromethymol blue will turn this beautiful, beautiful deep blue color, kind of like Levi's jeans. Sometimes it's called Prussian blue if citrate is the sole source of carbon and energy. So this is a Simmons citrate auger slant, and I'm going to inoculate the surface of the slant. This is a test that's run aerobically. Okay, so again, I'm going to harvest my E. coli. Okay, and then I'm going to inoculate my slant. Okay, start at the bottom and gently let the loop glide over the surface, kind of like in an S shape, like you're a snake slithering up the slant. Okay. And again, with all of these tubes, you guys, we want to make sure the caps are just barely loose so they can vent. Okay, I'm going to sterilize my loop. And folks, looking at the time here, there's one last workshop I need to go to get ready for going to online. So I think I'll only have time to inoculate the TSI slant, the triple sugar iron slant. So this weekend when we come back in to look at our results, I'll show you, with the movie showing results, I'll do the catalase test, which is a quick and easy test. So you guys, okay, so the TSI slant, this is not part of the IMViC test. This is a totally different test. And we're going to look for sugar fermentation and hydrogen sulfide production and molecular hydrogen and or carbon dioxide production during sugar fermentation. So this is a really fun test. So what we're going to do is inoculate TSI slants. They have 3 sugars: glucose, lactose, sucrose. They have the pH indicator, phenol red. They have sulfates is terminal electron acceptors for anaerobic respiration, and they have lots and lots of amino acids, including cysteine. So to run this test, you guys, we have to force our microbes to grow anaerobically in the butt. So we're going to use our inoculating needle, and we're going to do a stab and streak because we need our guys to grow both anaerobically and aerobically. So I'm going to harvest my E. coli. All right, and then I'm going to stab the butt. I'm going to try not to hit the bottom as I stab it, because that might provide an alternate way for oxygen to reach the microbes in the bottom. And I withdraw, and then, folks, that's the stab, and then I'm going to withdraw, and if I can see here, can't see very well, I'm going to streak the surface of the slant so that my microbes are also growing aerobically. Okay, that was a little bit awkward. Sorry about that. Oh my goodness. Okay, and again, folks, loose caps, we're going to incubate for 48 hours, and then we'll have a follow-up video showing you the results of all of our plates, all of our tubes. So that'll be kind of a fun movie to make this weekend if all goes well. Okay, folks, thanks for your patience. We'll have PowerPoints that will go along with these videos. Probably what I'll do is do the PowerPoint, and that way we can show you good pictures, provide more explanation, and then I'll probably maybe make these little movies as links, YouTube links, within the PowerPoint so you can actually see how things are being inoculated. Okay, and hope you all are doing well. Hope you all are staying healthy. Right, that's number one. Okay, take care, folks.